I'd like to thank Neil and ACAMP as well for giving me the opportunity to talk about Delta Genomics. So Ken mentioned that there might be three main things in technology commercialization that you should focus on. Focus being one, revenue, and speed. I'll try to touch on those in my presentation as well, because I agree with them. So basically a little bit of background. As Ken mentioned, I have a PhD in biochemistry and MBA degree. I started here at Delta Genomics about a year ago. One thing I don't come from is an agriculture or livestock background. That said, I work with a lot of researchers at the University of Alberta, and in particular Livestock Gentech, which is an institute at the University of Alberta focusing on on developing genomic technologies for the livestock sector. So Delta Genomics spun out of Livestock Gentech and University of Alberta for the purpose of offering the industry the services that have been developed at the lab. We are the first full-service genomics company specializing in livestock, in the livestock industry. What full-service genomics means to us means we can identify a single genetic marker, and we can sequence an entire genome to identify all the newest markers out there. Our goal is quite simple. It's to increase the profitability, competitiveness, and sustainability of the livestock industry. So I'm going to back up a little bit, and I'm going to talk a little bit about the market. So when I entered this, I didn't know anything about the livestock industry and why it was so important. And if we kind of look at the world population, here's a graph showing it from about 500 BC. The black line being what we estimate the population was, because we didn't have that great of records. The blue line being what the actual population is, and the red, orange, and green lines meaning what the population may be in the future. As we all know that we are beginning to overpopulate the world possibly, and what happens in the next 50 years will dictate whether this line continues to increase or decreases. And one of the main things which is going to affect that is the supply of food. To give you some other numbers, in 50 years it's estimated that we're going to need 100% more food. We all know what's going on in the world. We don't have 100% more land. We don't have 100% more resources to sustain that, which means we have to become more productive as in agriculture. And what I'm going to talk to you a little bit about today is how we're going to become more productive in the livestock industry. So a little bit of background. Since we first started domesticating livestock, up until about the 1700s, all genetic improvement in the livestock that we saw came from natural selection. We have different breeds of livestock associated with, up to that point, associated with regions. So we have animals that have adapted to tropical environments and animals that adapted to cold environments. It was not until the 1700s when someone by the name of Robert Bakewell came along and decided that there might be a better way to improve the genetics of animals. He's the first one to instigate line breeding and inbreeding for the purpose of producing livestock and animals that are better suited to meet the needs. So this marks the beginning of controlled breeding. And if you notice in terms of the world population, this is also the beginning of the Industrial Revolution when our population started to increase. So we as a society managed to increase productivity to meet the demand of growth. Or we might be say growth was facilitated by this increased demand. Which kind of brings us to an era of modern livestock breeding. So since Robert Bakewell and up to now, we've begun to implement more controlled breeding programs. We actively manage and measure traits. We developed tools such as crossbreeding, which is breeding two different types of animals to have an offspring to have more production, and it's become quite sophisticated. And just to give you some numbers, since 1920s, this type of breeding has resulted in increased milk production in cows by 300%. Hog finishing times have decreased 50%. Egg production in hen is up 90%. Cattle, beef cattle weaning and yearling weights are up 35 and 25%, meaning we're producing larger animals. So those numbers are impressive, but what do they actually look like? So here's an example of what improved productivity means in broiler chickens. Using 1957 genetics, this is what a broiler chicken looked at like in day 43, as opposed to day 85. Today, and this was even a few years ago, a broiler chicken at day 43 looks like this. A lot larger than it took us almost 90 days in the past. And after 85 days, this is what we have. Modern genetic breeding has really made a dramatic impact on productivity. When we look in the porcine industry as well, we've really focused on feed efficiency. So in 1972, it took 836 pounds of feed to produce a 220-pound pig. Now, in 2007, with 715 pounds of feed, we can produce a 275-pound hog. And what this means is that we're able to produce more meat with less resources. And we have to continue this if we're going to meet the food demand in the future. And so if you calculate, actually, I would have forgot I had this, but 300 pounds of feed reduction for 1,000 market hogs is actually 1,000 tons of feed a year. And so it is making a big difference. Which brings us now to the future of livestock breeding. Well, we're in a new era now, the genomics era. We finished the first deciphering the first human genome in just at the turn of the century, 2000. We now have genomic sequences for all the major livestock out there. And how are we going to use that to improve that? So first of all, a little bit background, what is genomics? Genomics is the study of an organism's DNA sequence or genome. It's the analysis relationship between genetics and its traits. So when it comes to livestock industry, we can link the genetic profile of an animal with the phenotypic traits, whether that's feed efficiency or growth or whatnot. It's a very powerful tool. And to give you a little idea of why we're calling it a genomics revolution, if we start looking at an example of a technology which we all know are familiar with, being computers, and we might have all heard of Moore's law. And basically, what this means is the power of computers has been exponentially increasing since the beginning. And we know this if we think back to our computers that we first had, the Commodore 64s, and the power now of computers. And it's a phenomenon which has been kept constant. If we apply that and we look at the cost of genomic sequencing relative to Moore's law, it looks something like this, meaning that in July 2001, it cost about $100 million to sequence a genome. With this chart here, in January, it was just over $10,000 to do a genome. The cost of sequencing is far outpacing Moore's law. And it suffice to say, this isn't even updated. As of today, we've actually hit the $1,000 per genome mark. So there is a revolution going on in the genomic space. So how do we apply this to genomics? Well, there's two main types. Something we call marker-assisted selection, which is a process to determine the genetic potential of an animal at birth, meaning we can do the genetics of an animal when it's born, and we can determine whether it's going to be highly productive or not. The other one is marker-assisted management, meaning that we could take a group of animals and we can say these ones have higher feed efficiency, so we should give them less feed to get to the same size as opposed to these other ones, and we can begin to manage our farms more economically. What that looks in reality nowadays, basically what we could do is we can take an animal, we can do a genetic analysis on it, and we can get, this is a very simplified version, information such as weaning weight, daily gain, dry matter intake. We can determine at a calving level whether that meat's going to have a high propensity for marbling or tenderness. And at a calf level, we can now begin to rank and sort these animals into groups to better manage them. And so basically what's Delta Genomics do? These are some of the things we're doing. We do sample processing, which means we're basically creating a biobank of samples, which means we are getting samples from all kinds of different livestock. We're storing them and recording the traits to give us an ability to do the genetics on this. And this is something which can become very powerful in terms of speed. This biobank is essentially a collection of, in the health side would be considered clinical trials. But in livestock, we don't have to go out and start a new clinical trial. Every time we have a new technology we want to test, we can store and bank samples, which is great for people such as yourselves inventing these tools, because we can quickly, and we get to speed now, apply those technologies to our biobank. And I won't go to too much details here, but basically we do marker-assisted management, marker-assisted selection, SNP profiling, which is marker selection, and we're doing this currently in bovine and porcine, and there's all kinds of panels out there for other livestock as well. And as I mentioned before, we can do full genomic sequencing, which is really for the discovery phase of the business. So I'm just going to conclude. I don't know how much time I've gone through, but I don't want to go too long. I'm about 12 minutes here. So in conclusion, there's perhaps been a perception out there in agriculture that, it's run by grandmas and grandpas, it's low tech, it's old, it's changing. That perception might still persist, but it's not the reality anymore. The reality of agriculture is it's a big business. It's highly competitive. It's driven by efficiency, cost, and innovation. And more, we believe that agriculture and livestock decision-making is going to be run through genetics and information we're getting out of the lab to help better manage. I'd like to just thank you for the opportunity, ask any questions, and these are some of the supporters. Western Economic Diversification funded the startup of our service lab. Livestock Gentech and University of Alberta is where we spun out of, and Alberta Innovates Biosolutions and Alma are big supporters of them as well. So thank you for having questions.
Any questions of Colin?
The database that you're collecting, which
Yes.
is important these days.
Yes.
Alone, just
Yes.
for tracking the cattle from birth to death.
Absolutely.
Yeah, so you're keeping that database, I assume, for a period of time, and you're able to track the animal?
Yes, and that's really one of the big part of the startup. So we've already have databases already. We have some that are over 1,000 samples with high density genotyping of them, and we have all that information tracked for that. And we're creating others. We just yesterday received about 15,000 samples from a feedlot, and each one of those samples has all the carcass data for that associated for each one of those 15,000 samples. So we can now begin to do some genetics on that, and we have that information that you can now begin to mine and control. So, yeah.
Last question?
Yeah, this was very informative, but my question is when you do all kind of genetic modification, and you, for example, in chicken, you can now consider chicken.
Yeah.
But what would the nutrient contain?
Sorry, the what?
The nutrient content?
The nuclear content?
Nutrient.
Oh, nutrient content.
Yeah. Would it also increase simultaneously, or we are increasing the size, but nutrient content is not increasing simultaneously?
Often, and yeah, we're increasing the size with reduced feed often, and we're not doing any sort of genetic modification here. We're just selecting for the animals that grow faster and eat less. And one of the things that the genetics does is it allows us to identify which animals will grow faster and eat less and mate them even before they've grown and eaten. It's a really powerful tool, but it's all about just enhancing what we're doing in modern breeding already. Sure. Yeah.
Just one quick question. So a lot of the feed efficiencies are determined mostly by the microbial community in the gut as opposed to the genetics itself. So are you also working on the microbial communities and inoculation to change feed conditions?
Yes, so some of the researchers that are associated with at the U of A are, in particular, the cattle with the rumen, which plays a major role, are doing exactly that. They're looking at the microbial content to see if that's going to play a role in feed efficiencies. That's one of the major projects that the researchers are working on currently.